Every human being, with the exception of identical twins, has a virtually unique genetic fingerprint. The chances of two people having the same genetic fingerprint have been calculated to be as low as 1 in 30,000 million, or 6 times the population of the Earth. Identification of an individual can be made with near certainty, and already this evidence has determined the outcome of many court cases. In Great Britain in January 1987, the Leicestershire police began a search for the killer of 2 teenage schoolgirls found raped and murdered 3 years apart in neighboring villages. Genetic fingerprinting of semen samples recovered as forensic evidence showed that both crimes had been committed by the same man. The prime suspect, who had actually confessed to the second murder, was eliminated when the DNA fingerprint of his blood failed to match that of the semen samples. In the following investigation, several thousand of the male inhabitants of the 2 villages were screened. The results were negative. Not a single blood sample matched the forensic evidence. But at a company picnic some 6 months later, a man was heard to say that he had provided a blood sample for his friend, Colin Pitchfork. Pitchfork was arrested, and in 1988 at Leicester Crown Court, he became the first murderer to be convicted by genetic fingerprinting. How accurate is genetic fingerprinting? Could it, or even should it, be used in a court of law to convict a person?
Take the Bible in your right hand and read aloud the words on the card.
I swear by Almighty God that the evidence...
It starts with the genetic blueprint. This is found in nearly every living cell, located inside the nucleus on the chromosomes. Each chromosome contains double strands of DNA. The information stored in a strand of DNA could be thought of in terms of letters, which spell out words, which are organized into sentences. And then finally, a series of instructions that help to determine what and who we are. In 1984, Professor Alec Jeffreys at Leicester University discovered that certain short sequences of DNA, words if you like, were repeated many times over. He discovered that these repeat, or core sequences, not only occur at many different locations in our DNA, but also that the number of repeats at each location varies from person to person. When processed and visualized with specific DNA probes, the core sequence produces patterns resembling barcodes. These are unique to each individual, hence the term genetic fingerprint. Like all genetic material, genetic fingerprints are inherited. Except for occasional mutations, each band in a genetic fingerprint is inherited from one or the other parent. It is a definitive identity and paternity test. The crucial aspect of genetic fingerprinting is that it generates positive identification. The technique was used to resolve immigration disputes in the United Kingdom as early as 1985. So how is the genetic fingerprint obtained? It begins with a sample of biological tissue. In many research and clinical laboratories, samples are routinely processed automatically. At Guy's Hospital in London, this research worker is using an automatic DNA extractor to produce a high-purity DNA fraction from a blood sample. First, cells in the sample are broken down to release the double-stranded molecules of DNA contained in their nuclei. A restriction enzyme is added to the DNA. This enzyme moves along the strand of DNA, cutting it at the so-called restriction sites. The restriction sites available to the particular enzymes used are located throughout the chromosome, except in sections consisting of copies of the core sequence. This produces large numbers of pieces of DNA, consisting of repeats of the core sequence. The number and length of these DNA fragments was determined by the initial distribution of core sequences in the chromosome. A small voltage difference is applied across the gel, and the fragments move through the gel at a rate proportional to their size. The gels are difficult to handle, and so the DNA band pattern on the gel is transferred to a nylon membrane by a technique known as southern blotting. First, the nylon membrane is placed onto the agarose gel. Then, absorbent paper is layered on top of the membrane. It acts as a wick, drawing up the DNA bands onto the membrane where they stick. Finally, the researcher adds a radioactive DNA probe, which will bind specifically to the core sequence. Excess DNA probe is washed off, leaving only the radioactive probe that is bound to the DNA pattern on the membrane. This can be shown on X-ray film, producing the fingerprint. We must now introduce a further level of complexity. There is not just one core sequence, but several closely related core sequences. Multi-locus probes will bind to all of these core sequences. This produces the familiar barcode with a large number of bars. A recent advance has been the introduction of single locus probes. These tend to bind to only one type of core sequence, producing a gel with two bands, one inherited from each parent. This does not give the same degree of statistical proof as the multi-band fingerprint, but by using two or more single locus probes, the statistics can be improved. The main advantage of single locus probes is their increased sensitivity, which makes them suitable for analyzing small samples of a sort often used in legal proceedings. These are called forensic samples. Genetic fingerprinting is a very powerful analytical technique, but like any laboratory procedure, it has specific limitations, and there are recognized areas where errors can creep in. We'll start by considering the method's limitations. First of all, the preparation of a genetic fingerprint uses a substantial tissue sample. A multi-locus probe will require several milligrams of blood or other tissue. Secondly, the sample must be in good condition. This can be a serious problem because the molecules of DNA are relatively fragile. They break down if the sample is exposed to heat or chemical reagents. If tissue samples are taken and stored under clinical conditions, then these problems won't arise. However, forensic evidence may be of such poor quality or present in such small amounts that genetic fingerprinting would not work. An innovation known as the polymerase chain reaction, or more simply, DNA amplification, solves both of those problems. It is so powerful that it is now possible to prepare genetic material from practically any biological forensic sample. The technique is simple enough to have been automated. and is now usually done using an instrument known as the DNA thermal cycler. The sample under investigation is first treated to dissolve its DNA content. The solution is then heated, which causes the double strands of the DNA molecule to separate. DNA polymerizing enzyme is added. This uses each single strand of the original DNA as a template to build up 2 new copies. So there are now 2 identical double-stranded molecules of DNA. A second heating cycle separates the 2 double strands, and the polymerizing enzyme gets to work again. So you now have 4 double-stranded molecules of DNA. With each cycle, the number of DNA molecules is doubled. Millions of copies of the original DNA molecule can be synthesized in a few hours. DNA amplification also has a direct clinical relevance. For example, it is now being used during prenatal screening and diagnosis. This pregnant woman is a carrier for the genetic illness cystic fibrosis. Her fetus can be diagnosed as early as 12 weeks by using a technique known as chorionic villus biopsy. Fetal cells are removed from the placenta. The DNA from these cells is amplified and used to produce the fingerprint. The extraordinary power of DNA amplification is both an advantage and a potential disadvantage. The technique is so sensitive that even trace amounts of extraneous DNA might also be amplified. Careful quality control is needed to prevent this from happening.
Now we've got a body of a female woman.
The contamination of samples must be carefully considered when gathering evidence from the scene of a crime such as murder.
As you can see, we've got...
In this reconstruction of a real incident, the police were called to investigate a murder. On arriving at the scene of the crime, they discovered that the lady's husband was also dead. Was it a murder and a suicide, or a double murder committed by a third party? Scene of crime officers and forensic specialists are trained to take particular care with samples such as bloodstains. Air drying and cool storage can be very effective in retaining DNA structure. The DNA in bloodstains can still be fingerprinted after a period of several years. However, a bloodstain could contain blood from both the victim and the aggressor. This could cause serious problems of identification. So in these situations, a sample is taken from the victim to provide an internal control. Any bands in the sample which were not present in the victim's sample can then be identified as the aggressors. In general, the utmost care has to be taken with the collection of samples, and they must be carefully prepared before fingerprinting or DNA amplification is possible. There are 2 categories of error which we should consider. Both were highlighted in the case New York versus Castro, 1987. First, there is laboratory error. Because of the complexity of the technique, samples might be mislabeled, transposed, even lost. Strict quality assurance procedures are necessary to prevent this. Even minor changes in the experimental conditions can result in misleading information, producing a DNA print with too many bands or bands in the wrong places. Standard protocols have been established in some countries, but there are no international standards of procedure. The second major category of error is observer error. For example, the bands in a fingerprint may be blurred, making it difficult to tell where one band ends and another begins. However, the human eye is amazingly good at detecting matches or mismatches in the multiple band prints produced by multiple locus probes. Human observation is still more effective than any of the automated scanning systems. Interpretation of single locus probes can be more difficult. How far apart do two bands have to be before they are definitely different? The statistics have not yet been universally agreed on, and this is another possible source of error. Problems of incorrect lab procedure or data interpretation are far more likely to make two identical samples look dissimilar than make two different samples look alike. The guilty may rarely get off, but the innocent are extremely unlikely to be falsely incriminated. Finally, there are errors of data interpretation. Suppose the forensic sample matches the lab sample. For the result to stand up in court, the expert witness must be able to calculate the chance of an accidental match and demonstrate that it is highly unlikely that the same pattern might have come from two different people. This depends on knowing the baseline frequency of the various genetic markers, not just in the general population, but also in specific subpopulations such as ethnic groups, where the frequency of any one marker may vary significantly. Much of the necessary information is available and is already being used in court. The United States Congress Office of Technology Assessment has appointed a panel of specialists to investigate these problems. When a virus binds to the surface of a cell, the viral genetic material makes its way to the cell's interior. DNA analysis can identify these traces of viral DNA before symptoms of the disease appear. AIDS can have a latent period of several years. It is thought that only a very few cells, such as this lymphocyte, actually carry the virus, shown here as small green particles. DNA amplification and analysis is powerful enough to detect these minute traces of viral DNA. It is capable of detecting less than 10 copies of viral DNA per million cells. In cancer research, the vast international databanks of autopsy material can now be used to trace the contributions of viruses and mutations to cancer. In population studies, the presence of genetic markers in tissue samples hundreds or thousands of years old can help anthropologists to map out the migration of ancient peoples. In veterinary science, genetic fingerprinting is increasingly used to establish the pedigree of a wide range of animals, from Old English sheepdogs to stud bulls. In agriculture, the breeder's investment in genetically improved crops or livestock can now be copyrighted. Genetic fingerprinting means that every cow, every ear of corn, can be identified and traced back to its rightful owner. Genetic fingerprinting is already changing our lives and will probably come to be considered one of the greatest discoveries in modern science.